So today is great honor to have Dr. Gordon Freeman to talk about cancer immunotherapy. Welcome.
Thank you for the amazingly kind introduction and the invitation by Biocytogen. So these are my disclosures. Now, we can now unleash the immune system to attack cancer. And how did we learn to do this? And really it was by coming up with a new strategy, which was to stop the tumor from turning off the immune response. And that's been termed checkpoint blockade. Now, I came into studying the field of immunology by studying co-stimulation, the second signal of B7 through CD28 that gives you optimal T-cell activation and expansion. Now, as the human genome began to be sequenced, we asked what other molecules looked like the B7 molecules. And we identified a molecule that we now know as PD-L1. And working with Clive Wood and Tasuku Honjo, we showed that PD-L1 was a ligand for PD-1. And the interaction of PD-L1 with PD-1 led to tyrosine phosphorylation of the cytoplasmic domain of PD-1, activating and recruiting the SHP2 phosphatase, which then dephosphorylated proximal signaling molecules in the T-cell receptor CD28 signaling pathways, leading to reduced T-cell proliferation, cytokine production, and cytolysis. Now, if you block this pathway, you can unleash an anti-tumor immune response with increased cytokine production and T-cell cytolysis. Now, this has been translated into human clinical trials, and PD-1 or PD-L1 antibody drugs are now approved for 25 different tumor types, representing about 50% of cancer deaths. The response rates can range from about 90% in Hodgkin's lymphoma. The common solid tumors are in the 20 to 30% range. PD-1 therapy doesn't work in certain tumors, such as pancreatic or glioblastoma or standard colorectal. So there's tremendous opportunity to bring further benefit to patients. So for many years, I've put up this slide that the future is combination therapy. And indeed, there have been some notable successes, such as PD-1 combinations with CTLA-4 or LAG-3, or the cisplatin drugs that lead to immunogenic cell death, or PD-1 with angiogenesis blockade. But there have been several early failures, such as IDO or MEK inhibitors or CSF1 receptors. And more recently, there's been an increasing number of failures, such as TIM3, CD47, TIGIT, TGF-beta blockade, or STING, that we really had high hopes for. So there's really now a reassessment of combination therapy. And what do we have to do to prioritize the over 300 agents that have shown combination activity in a mouse model and accurately and efficiently translate them to human therapies? So I think what I would say is that we need to model the combination therapies with PD-1, PD-L1 therapies in mice more accurately. In particular, I think the PD-1 antibodies that we use in mice have to have characteristics similar to Pembro and Nivo. Now, of course, they have to block receptor-ligand interaction, and indeed all the anti-mouse antibodies do. They need to have high affinity, and indeed, that's where there's some failure. The RMP-114 antibody that's often used in tumor immunotherapy actually has about 100-fold lower affinity than Nivo or Pembro. We also want to use antibodies with an FC with low antibody effector activity, such as the human IgG4 used in human therapies. We also want to use same species, so a mouse anti-mouse, so that we can dose for extended periods, as in humans. The other thing is we don't want to use low doses of PD-1 antibody. And some investigators, to get a publication, will use a little bit of PD-1 plus their combo agent, basically tipping the scales for combination efficacy. And we don't want to use low affinity PD-1 antibodies. Now, alternatively, we can use actual Pembro or Nivo in a human knock-in PD-1 mouse. And also, we should use tumor models that are resistant to PD-1 therapy, because in the current clinical world, the clinical trials are often going to be with patients who are not responsive to PD-1 therapies. The other thing we need to do is design the trials in the best possible way. And indeed, Mike Atkins and Mario Snoll have really presented better ways to design the phase 3 trials. And this is published in JITC, and I'd highly recommend looking at this in detail. Just in brief, what they recommend is that the mechanism of action and the biology should be additive or synergistic, and that it should be tested in valid preclinical models and have single agent activity. In addition, the phase 1 and 2 data need to be more rigorous. Again, the agent needs to have single agent activity in early clinical studies. Really, one thing they emphasize is that the phase 2 trials be randomized. They should look for activity in standard of care tumors and PD-1 resistant tumors. And also, ideally, the therapeutic responses are durable, and the agents have non-overlapping toxicities. The phase 3 trial designs should be good translational studies with an integrated biomarker program, so that you can really, whether the trial fails or succeeds, you can understand what happened and gain benefit from the knowledge. Now, what I wanted to next talk about is how can we identify new checkpoints which are non-redundant with the PD-L1, PD-1 pathway. In particular, that can be targeted for kidney cancer immunotherapy. And this is the team investigating HHLA2. So a number of immunomodulatory B7s can be expressed by tumors. And we were particularly interested in a molecule called HHLA2, which was known to have one stimulatory receptor, TMIGD2, but the studies with the Fc fusion protein had shown that the dominant biology was an inhibitory immune effect. Actually, the family investigations actually continue, and our son, Sam Freeman, did the bioinformatics of this analysis. And basically, we're looking at the expression of B7 family members in renal carcinoma. Basically, what you can really highlight here is that HHLA2 expression is moderate in normal kidney, but greatly increased in kidney cancer. PD-L1, little changes. PD-L2 goes up a bit, B7H3 more, B7H4 goes down. But this really focused us in on this HHLA2 molecule. So we wanted to understand what was the receptor leading to immunoinhibition. So we did use the HHLA2 FC to screen 5,000 cell surface proteins with retrogenics and identified KIR3, DL3, as a specific receptor for HHLA2. HHLA2 didn't bind any of the other 15 KIRs, just the KIR3DL3. Now, the KIR3DL3 has a single ITIM in its cytoplasmic domain and 3 IG domains consistent with an immunoinhibitory function. And it's relatively rarely expressed. It's on some intratumoral T effector memory RA cells, some activated T cells. It's on the CD56 DIM NK cell population. Now, the HLA2 binding to KIR3 inhibits T cell and NK cell activation, published by us and also by Xingxing Zhang's group. So we got together and formed a biotech startup, Next Point, to try to explore this pathway for therapeutic applications. We showed that engagement of the KIR3 by HHLA2 inhibits T cell activation and IL-2 production. Here, using a Jerkit CHO cell antigen presenting system, we can see that if you add HHLA2 expression to the system, that we basically turn off IL-2 production. If you look at NK cell lysis, you can see either KIR3 or anti-HHLA2 antibodies. Here's control antibodies. Here's the blocking antibodies. You see greater NK cell lysis with blockade. We found that HHLA2 was expressed in over 70% of renal cell cancers, as indicated here. Nice expression on the cell surface. It's not expressed on all RCC. And if you look at the TCGA database, you see certain high expression of HHLA2 in colorectal, lung, pancreatic, and renal cancer. And indeed, multiple groups have now reported that high HHLA2 expression is associated with worse outcome in lung, colorectal, cholangiocarcinoma, lung, and tongue cancers. Now, the thing that really fascinates us about this pathway is how non-redundant it is with PD-L1. If you look at the immunohistochemistry, and each dot indicates an individual tumor, you have roughly 20% of renal cancers which express PD-L1. And you have probably about 50-60% that express HHLA2. What you don't see is there's no dots up here. There's no co-expression. So co-expression is really rare in renal cancer. And our colleague Xingxing Zhang has also shown this same pattern holds in lung cancer. Now, if you look at single-cell RNA-seq with David Braun, the same pattern shows. You get individual tumor cells which will express PD-L1 or HHLA2. In contrast, if you had co-expression, you'd see a pattern like this, such as seen for CD8. Now, the HHLA2 is regulated very differently than PD-L1. Gamma interferon is a wonderful upregulator of PD-L1, but not HHLA2. So here you can see gamma interferon, TGF-beta, IL-10 don't upregulate HHLA2, whereas gamma interferon strongly upregulates PD-L1. Now, if you look in vivo RCC tumors, what you see is that if you take a tumor explant, you see HHLA2 expression. It's fairly quickly lost over the span of about 2 weeks. The same pattern is seen with RNA expression. Now, if you take an RCC cell line, most of them don't express HHLA2 in vitro. Put it back in vivo, in this case in an immunodeficient mouse, and it reacquires HHLA2 expression. You can see by immunohistochemistry also. Take it out of the mouse, and it quickly loses expression. So the regulation of HHLA2 expression is provided by an in vivo signal. It's not hypoxia or a wide range of tested cytokines or culture conditions. It's not dependent on an adaptive immune signal, since it happens in an immune deficient mouse, and we're investigating the mechanism further. Now, in vivo, survival after PD-1 therapy in RCC, in this case nivolumab, is best in the PD-L1 high HHLA2 negative tumors, and it's worst in the tumors that express both PD-L1 and high HHLA2. So we think this is an immunosuppressive mechanism for tumor-resident T and NK cells, where there's immunosuppression via this pathway in the tumor. So there are challenges in developing agents here because the KIR expression is relatively rare, and this whole pathway, the receptors and the ligands, are expressed in many mammals, but not in rodents. So there's not going to be an orthologous mouse model, and we have to model it in humanized mice. Now, our colleague Xingxing Zhang went ahead and heroically did this, transferring in an HCC tumor and human NK cells, propagating the human NK cells with IL-2. And when he did that, what he saw was that the blockade with a KIR3 antibody led to reduced tumor growth. So we think this can work in vivo. And NextPoint now has made agents blocking the pathway, either HHLA2 or KIR3DL3, and is in phase 1 trials with both agents. So in summary, the KIR3 HHLA2 is an immunoinhibitory pathway. Blockade can enhance T and NK activity. It's expressed in the majority of RCC, and it's non-overlapping with PD-L1. And we hope that we'll have monotherapy activity and be relevant in a proportion of PD-1 refractory tumors. Now, shifting gears, I wanted to talk about some of our work with the microbiota. I feel cautious about presenting this with Dan in the audience. But, and Dan and I go a long way back. We were talking when he was isolating CD8 and CD4, and I was isolating T-cell activation genes. So how does the gut microbiota regulate the anti-cancer immune response? And how can this be used to identify new inhibitory pathway targets for immunotherapy? Now, this work is a collaboration between 3 labs. Dennis Kasper is providing the microbiome expertise, Arlene, the immunological expertise, and my lab, the RGMB PDL2 expertise. The people who really did the work are Junshu Park and Francesca Gaznaga, and Yanping Xiao started the RGMB work and is in the audience. So RGMB, she showed, was a ligand for PDL2. RGMB stands for repulsive guidance molecule B. It's a member of a small 3-gene family, first identified in the nervous system, and it's a GPI-anchored protein. Yanping shows the RGMB PDL2 interaction would regulate respiratory mucosal tolerance. Now, this is a pathway that intersects multiple pathways. We have the classical interaction of PDL2 with PD-1, recruiting phosphatases and turning off T cells. Now, with RGMB, we have bone morphogenetic proteins, BUMP2 or 4, and RGMB can act as a co-receptor carrying them to the bump receptors, engaging MAP kinase, ERK pathway, and SMAD activation. And PD-L2 may intersect here. The RGMB also can dimerize and cross-link two neogenin molecules, engaging Rho pathway and cytoskeletal rearrangement. And how PD-L2 intersects this is still under investigation. So we looked at how this intersects with the microbiota-dependent models for anti-tumor immunity. Now, basically, the guts of mice were cleaned out with a 4-antibiotic cocktail, and then a human microbiota generated by the Caspar lab was added back to the mice after antibiotic treatment, reconstituting with the human microbiome. Tumor is implanted and treated with PD-1, PD-L2, or other immunotherapy agents. Now, as Dan showed you, if you antibiotic treat a mouse, there's no longer a response to anti-PD-L1 or PD-1 antibodies. But if you add back the microbiota, then the mice become responsive again. So the human if you look at the T cell and myeloid populations in this mouse, in the responder mouse, the one with the human microbiome, there's increased CD8, CD4, and CD11B and 11C cells in the draining lymph node. Now, does the gut microbiota modulate expression of co-stimulatory or co-inhibitory molecules? And what we saw was that in the non-responder, the antibiotic-treated mice, there was a high expression of PD-L2. But if you added back the human microbiome, there was a reduction in PD-L2 expression. The same was seen with the mouse microbiota. So does higher expression of PD-L2 inhibit anti-tumor immunity in these mice that aren't responding to PD-1? And we asked, can PD-L blockade overcome this lack of efficacy? Now, remember here, if the PD-L2 blockade is important here, it's probably not working through PD-1 because the PD-1 antibody alone doesn't work. So what Francesca and June saw was that if you treated the antibiotic-treated non-responder mice with PD-L1 and PD-L2, that they responded. The single agents didn't work. So the combination did. So now does this blockade of PD-L2 is the important receptor PD-1 or RGMB. And again, Yanping had identified this pathway. So the question is the PD-L2 blockade working through the PD-L2 mediated PD-1 signal or through the RGMB signal? And Yanping had made antibodies that would block only the RGMB PD-L2 interaction or would block the interaction with both PD-1 and RGMB. So a single or a double blocker. And what we found was that the either PD-L2 antibody worked, either the single blocker or the double blocker in combination with PD-L1 would reduce tumor growth. So this says that it's sufficient to block the PD-L2 RGMB interaction. Now, what if you block the other side? What if you block the RGMB? And so we had antibodies that blocked RGMB, and indeed, the combination of PD-L1 plus RGMB, or PD-1 plus RGMB, gave better tumor control than either agent alone. Now, is this working by cellular depletion or just blockade? So we made an Fc effectorless form of the RGMB antibody, with the LALA-PG mutation. And indeed, the effectorless RGMB antibody gave benefit when combined with PD-L1, actually a little better than the wild-type antibody. So it's working through blockade, not cell depletion. So where is the RGMB expression important, on T cells or on APCs? Most studies have really emphasized RGMB expression on myeloid cells. Surprisingly, when Arlene's lab looked at conditional RGMB depletion in T cells or myeloid cells, what we found was that if RGMB was present, that the combination, that PD-L1 therapy didn't work. But if you removed RGMB just on T cells, then PD-L1 antibody would work, indicating that RGMB expression on T cells is what's important. In contrast, if you knocked it out on myeloid cells, it didn't make a difference. So the RGMB expression the normal microbiota would reduce RGMB expression on CD8 T cells. Now, can we identify a single bacterial species in human microbiota that can modulate PD-L2 expression? And would it replicate these biological results? So the goal was to identify a single bacteria, and the Caspar lab used individual antibiotics, and they basically, this slide says that there's a gram-positive anaerobe from human microbiota that can promote anti-tumor immunity. They then plated bacteria in isolated individual colonies and really focused in on a Clostridium subtype species called Coprobacillus catenaformis, which is a gram-positive anaerobe. And mice were monocolonized with just this one C. catenaformis bacteria. And what you see if you colonize with just the one bacteria is you reduce PD-L2 expression on the myeloid cells. And if you look at tumor immunotherapy, the mice which are antibiotic treated and don't have the CCAT are non-responsive to PD-L1 antibody. But if you monocolonize it with CCAT, then they become responsive. Now, we don't at all think this is the only bacteria can do this, and we don't at all think this is the only mechanism of immunoregulation that the microbiota has for regulating anti-tumor immunity. But we think it's novel, and the Caspar lab has found that an extract from cell wall can replicate this effect and is trying to identify an agent that can do this. And indeed, we've formed a biotech called IOM, which is trying to advance these therapeutics. So in summary of this part, the PD-L2 RGMB axis may be a key mediator of tolerogenic interactions between the microbiota and the host immune system. A normal gut responder microbiota may downregulate PD-L2 and RGMB expression, and in non-responder situations, such as germ-free or antibiotic-treated mice that don't respond to PD-1 or PD-L1, these have upregulated expression of PD-L2 and RGMB that is functionally significant. Now, blockade of this pathway or conditional deletion in T cells overcomes the microbiota-dependent resistance. And it really identifies therapeutic combination possibilities of PD-1 plus RGMB antibody, PD-1 plus PD-L2, PD-L1 plus L2, or bispecifics, which you'll soon hear about, as therapeutic combinations. So these are the people in the lab who've done the work over the years. And this is a wonderful group of collaborators who've worked together on the B7 CD28 family pathways for over 35 years. Thank you.